Hello, everyone. I want to welcome you to our HHRF, or Horses and Humans Research Foundation webinar. And tonight we're going to be talking about making sense of horses and humans interaction, exploring the real world data. And we have Dr. Laurie McDuffee, who is a veterinarian, but she's also teaches at University of Prince Edward Island, and she's a large animal surgeon. And we have Dr. William Montelpare, not Montelpare, sorry. I thought I had it and then I changed. And you said last time that you really kind of call yourself a professor of wellness. Is that your intro?
I'm a research chair, so I am the chair of human development and health. So it's all about wellness, yes. I'm a biostatistician, yes.
But you go by Bill.
I do. I go by Bill.
Okay. All right. So I'm going to turn it over to the both of you so you can tell us about your research.
Great.
Thank you, Pebbles. Welcome, everyone, and thank you for joining us. And we'll go ahead and get started. Even though this webinar is focused on exploring research data, we try to keep a real world perspective. As Pebbles said, I'm a veterinarian and a surgeon. I also do research and I teach vet students. But I think of myself as a clinician, and I'm always looking at our research through a clinical lens.
So many of you would have called the stuff we're going to talk about tonight reporting data from field work. However, the term that we'd like to use is this is real world data. So we've taken a number of scientific principles and applications of research methodology, and we've brought them together this evening so that we can actually make sense of horses and humans and the interaction between horses and humans. Oh, sorry, I went to, there we go, back one.
There we go. Okay. In this webinar, we will discuss what we've learned from several of our different research studies where we've had horses interacting with humans, and we've collected the data in these studies primarily from the perspective of the horse. One of our guiding principles of our research has been to evaluate the welfare of horses under different conditions, which have included participating in research studies as participants in therapeutic riding programs, as patients undergoing various veterinary procedures, and as participants in equine facilitated psychotherapy, which we presented in the last webinar last month. Evaluation of well-being in these studies has primarily focused on analysis of stress. And so we will be focusing on describing different ways that we have processed or have been processing measurements that we considered in understanding stress during interactions between horses and humans.
So in the beginning, we asked the question, what is stress? And we found out that it's really hard to measure stress directly. In fact, we say we cannot measure stress directly because stress is a construct. And here we show a fairly complex diagram of the physiology that underlies stress. And that's part of the basis of what we're going to be talking about this evening. For the most part, stress is a latent variable. In other words, it's a hidden variable. And as such, we needed to figure out a way that we could measure stress under different levels of experimentation and with different levels of precision. So in our case, we measured stress with a behavioral scale, and then we measured stress with a physiological systems scale, that it being heart rate variability. And again, here we have some great measures of time domain and frequency domain, and the measures of entropy using the nonlinear approaches of the point care plots. And then we measured stress with the hormonal responses of oxytocin and cortisol under different experimental conditions.
Oh, good. Oh.
Yeah, sorry.
The slideshow has a
It's going to be tricky. There you go.
mind of its own, I guess. So which of these is the best measure? During our research program into human-horse interactions and stress responses, we've been trying to answer this question.
So delay, I think. There you go. Yeah.
So we're going to walk you through some of our projects and how we have progressed in our evaluation of and our understanding of stress responses in horses. The first study was an interesting study. You might have noticed from my bio that most of my research career has been studying bone healing, fracture repair methods, and stem cell research. And so more recently in my career, I took an interest in equine behavior. And this was one of the first studies that I did with an honors student. And it was a comparison of training methods for horses that had aversions to oral medications. Okay. So we had horses and donkeys that were chosen based on displaying stress behaviors during administration of oral medications. I was doing some positive reinforcement for horses in the veterinary teaching hospital that were averse to different medications. And in this study, we went out in the field and we found owners who had horses that they couldn't medicate orally. And these horses, so the behaviors were significant enough that the owners could not deworm their horses and they couldn't treat them with oral medications when the vet asked them to. So our research objective with this study was to determine if one behavior modification method was more effective than the other. I was interested to find out, should I be using positive reinforcement only or negative reinforcement? Was it just as good? We wanted to find out if one of these methods was more effective than the other. And at the same time, we wanted to be sure that the most effective method was not causing more stress. So here, image one, they look pretty similar, but image one is actually my student using positive reinforcement. And so as a refresher, positive reinforcement, we added a little food pellet when the correct behavior was displayed. And that correct behavior was the horse keeping their body and head quiet. So of course you have to do this in baby steps. Each session would have a different step towards the ultimate goal of the horse quietly accepting oral medications. And image two demonstrates negative reinforcement, where we remove an undesirable or unpleasant, or in this case, aversive stimulus, which was simply the dose syringe. As soon as the horses saw that, they did not like it, and they started their aversive behaviors and stress behaviors. So in negative reinforcement, when they did the right behavior, we removed the dose syringe. So this was our first foray into using heart rate monitors and behavior scoring as a way of evaluating the stress responses of horses involved in experimental research. As I said, I'd been doing a lot of stem cell research, but I came back from an International Society of Equitation Science meeting where there were quite a few posters talking about heart rate variability and stress. So I came home and I thought, well, I'm going to buy some polar heart rate monitors and we're going to give this a try. So it was our first foray. And so we did. We bought the heart rate monitors. We instrumented the horses and donkeys during their behavior modification sessions. And primarily we collected heart rate data as a measure of stress, and we recorded all the behavior modification sessions. And then after the fact, behavior was rated by 2 equine experts using a 4-point ordinal scale, where 1 equals no stress and 4 was severe stress.
Okay, so in regards to, did I jump over?
No, you're good.
Okay, sorry.
In regards to.
Okay, so in regards to measuring effectiveness here, there was no significant difference between the measures that we observed in terms of the number or the duration of the sessions. And in regards to measuring stress specifically, there was no significant difference in the heart rates between the groups. So again, this is not heart rate variability yet. This is just looking at heart rates.
Yeah.
Have I jumped over?
Yeah.
That's what I thought I did.
Yeah.
Okay, there it is there. So we didn't see any differences there in our ANOVAs and the pre-post training. Sorry about that delay in our jumping. Okay.
Stress rating.
So with the stress ratings, there was no significant decrease with increasing the number of sessions. However, there was also no significant difference between the groups that we were measuring as well. So on both accounts. So it indicated that both methods were effective and both methods were humane. In other words, both of the behavioral interventions were doing what the researchers intended to do. Okay.
This was really a pilot study.
Is that this here?
Yeah, you're okay.
Okay. Sorry about this. Our slides are not jiving with our screen. Okay, so this was really a pilot study for using heart rate monitors and behavior and to measure our stresses. And so we learned a lot about it. We learned a lot about the applications of the heart rate monitors. We learned a lot about how we could measure stress. And we started to talk about different ways that we could actually consider some of the measures using objective versus anecdotal measures that are typically used. So we started to explore heart rate variability as one of our modus operandi. So I'm also a human physiologist, and at the time, I was using heart rate monitors to measure heart rate variabilities in humans under different conditions. And so this was not just my first foray into working in this area at all. It was completely my first foray into working with animals and applying human physiology principles to veterinary medicine, which actually raised a number of questions, but all good questions.
I had to bring him over to the burn.
Yeah, I went over to the dark side.
Teach him how to approach a horse.
Yeah.
Anyway, in study number 2, we began to focus on the use of the heart rate variability as a viable measure of equine physiological responses. And then this led us to ask about the stability of the polar equine heart rate monitor as a tool when used in equine research under different conditions. Heart rate variability has been used extensively in studies of humans and horses and other animals, of course, to evaluate stress.
And so the purpose of this study was to measure test-retest reliability. As a biostatistician, which is my primary occupation, I was really concerned whether or not we were actually able to get the same kind of responses in equine participants as we were in humans. And in fact, the research I was doing at the time was testing the stability of the test-retest measures in humans. And so we took that same concept over to horses to measure heart rate variability in horses. And so it really seems a bit of an oxymoron, right? We're trying to measure stability in a measure that is intended to be variable, but it's not a chaotic measure. It's just that it's a variable measure of what's happening in terms of the heart rate responses. So
in this case, so for this study, we went out in the field and we had 32 horses at a couple different stables, and we instrumented them in their normal environment with the heart rate monitors. And we took them through 3 different activities. So we had them free in their stalls, and then we had them confined on cross ties, and then we had them being led at a walk.
Jump over.
Go.
Okay,
Oh,
so,
no.
yeah, okay, there you
Yeah.
go. So one of the ways that I always promote measuring reliability is using the intraclass correlation coefficient, because it provides us with a case-specific test-retest measure as opposed to the Pearson, which is a group measure of responses. And so here we show the stability of the heart rate monitor under those different conditions, cross ties, stall, and walking, using time domain and frequency domain measures. And so the measure that we're looking for is a high intraclass correlation. And you can see that under certain conditions, we did have high intraclass correlations under the time domain measures for very specific activities. We saw high intraclass correlations, but we saw lower intraclass correlations in the frequency domain measures than we did in the time domain measures.
I have a question in the chat box real quick.
Okay,
I would like you to define HRV, and is it beat to beat or changes?
It was heart rate variability was our measure of the distance between R to R waves. And so if you consider the PQRST of an electrocardiogram, the R wave, okay, so it goes P and then Q, R, and then ST wave. So the R to R distances is what we were measuring. In fact, if you look here, you can see the R to R distances. Okay, and so from the R to R distances, we actually measure several other derivatives. In other words, under the time domain, where we're measuring the x-axis being time, we measure the root mean square. We measure the standard deviation of the normal of 50 normal beats in there, so SDNN, and we measure heart rate, and we measure the mean distance across those measures. Okay, when we look at when we look at the frequency domain, we're talking about the frequency of frequencies. In other words, how often does a particular count happen? Okay, because a frequency is a count, and we're counting the number of counts of a certain type. So how often do we get 5 counts? How often do we get 4 counts? How often do we get 3 counts? And from there, we get derivatives as well that give us an indication of the power of the stimulus relative to sympathetic and parasympathetic systems. And so in that case, one of the measures that's commonly used is the low frequency, so the sympathetic drive, a big blast of acetylcholine to stimulate the heart and fight or flight, versus the parasympathetic, the low, many high frequencies of a count that is under the parasympathetic system. So that's what we're looking at in these initial measures. And we wanted to see how consistent were those measures from time 1, day 1, to time 2, measured 2 weeks later. Okay, I hope that answers the question.
Yes, thank you.
Okay, cool. So what's really cool about this is we started to use a lot of data visualization to make sense of this, because again, even the intraclass correlation it's an analysis of variance technique. It's a mixed model. It gets a little bit hairy in the math. So what I wanted to be able to do is show what are the consistencies in the responses of our animals under the different conditions. And so this is a great graph, because this is actually looking at a group of horses. These are the standard bred horses.
Well, these horses, we had a mixed,
Here.
different breeds. Actually, standard bred horses at the track don't walk very often with someone leading them. They just walk with them. So, in fact, we don't have them being led by somebody here, but all the quarter horses and other sport horses are on this graph.
Yeah, so that's what it was, the quarter horses. So in this case, you'll notice, and I can use my mouse here, notice here, this red line represents the responses of each of the individual horses in the cross ties. So I know it's a line graph, but it's the principle that I'm trying to show you. Normally we just have dots, but it's a line just to show you the differences here. And the blue graph represents the response of the horses in the stall. So notice this particular horse, number 24, is very consistent in its responses during the stall and during the cross ties. But look what happens during the walk. And this is consistent for all of the horses. The walking data in the heart rate monitor is not matched up with the stall and the cross ties, which now it pauses us. It makes us think, wait a second, what is happening in this data? So if you're doing a study and you're trying to demonstrate the stability of heart rate variability or any kind of measure, you now have to be cognizant that the data you get for an activity matters. And in this case, we're showing that the data you get in the stall and the cross ties is not the same as the data you get in the walk. And so that's extremely valuable information for us, especially as we're trying to demonstrate the stability of a measurement that is actually variable. Okay? So now we started to say, okay, you know what, this is really exciting from a complete nerd advantage here. We started thinking, what's going on with these data? And so there's a technique referred to as the BlandAltman measure of agreement. And I'm not sure if you know this, but it's a clinical technique that's used. If I had a clinical device or a clinical approach and I wanted to demonstrate, and I consider that device the gold standard, then I'd like to do, and I'd like to invent a new technique or a new device, and I want to compare it to something, I would compare it to the gold standard. So with the BlandAltman approach, I can look for agreements between measures or between different events. And in this case, we're showing the agreement or the consistency of the responses of the data for each horse. In this case, it's with stall and cross ties. So now I'm using a statistical methodology to support the conjecture that things that happen in the stall similarly happen in the cross ties, but they don't happen in the walk. And you can see here that there's a lot of agreement, except for one outlier that falls below the lower boundary of the characteristics that I set for the parameters of the BlandAltman approach. And what I'm trying to show here is that there are data visualization techniques that help us to explain the phenomena of what's happening during these particular events. So what we concluded from this is that heart rate variability is statistically stable during specific activities, okay? But heart rate variability is complex. And it's complex because it's providing us with information about the two complementary systems of the autonomic nervous system. That is the parasympathetic and the sympathetic system, which we talked about in our last webinar, but again, that the sympathetic system being the fight or flight system and the parasympathetic system saying, whoa, relax. And so we know that the heart rate variability is influenced by the intrinsic characteristics of the person or the horse in this case, and the extrinsic stimulus or the environmental stimuli around us. And so hold that thought, because it's the environmental stimuli around us that really is important to where we're going in our research. So heart rate variability is good, even though it's variable. We know it's statistically stable, so it provides a measure across different measurement domains, both time domain, frequency domain, which are common, and then the nonlinear point care plots, they show the expression of entropy or the randomness or the variance within the measure. So we can use that information to our advantage to make sense of these data. Okay, here we go. There we go.
Okay, so our third study that we're going to talk about was an observational evaluation of stress in horses during therapeutic riding sessions. Bill and I actually volunteered for a Canadian Therapeutic Riding Association therapeutic riding program. And we were interested to find out, you know, these horses are so well behaved, but were they still stressed? So this study, we used similar measures of interest. We did look at heart rate variability across the 3 domains. And we also collected saliva to get cortisol levels. And we were going to use stress behavior counts. And we collected the data on therapeutic riding days during 3 activities while they were free in the stall before any session started. We called that baseline. And during 2 activities that we thought should be stressful if it wasn't, during the mount and during the dismount, because of course, most of the riders in these programs have to have different mounting type systems that they use, adaptive mounting and dismounting systems. So in this case, one thing that was a little bit different from the past was using a stress ethogram to help us determine levels of stress and to be more specific than simply asking equine experts, does the horse look stressed or not? And so we wanted to go beyond providing simple ratings of stress to using the ethogram. Now, most of you probably know this, but an ethogram is a catalog of behaviors with an operational definition. And the operational definition makes it clearly defined so that by rights, anyone could watch the videos of the horses and determine which behaviors were being displayed because they're so well defined. Now we used an ethogram that was previously published as a stress ethogram for horses in therapeutic riding programs. And we modified that published ethogram slightly to make sure that it fit our horses. Okay. And one of the ways we wanted to compare the stress behaviors was by creating these radar graphs. The radar graphs are valuable because they show us the frequency of a given type of stress behavior from our ethogram. And so you can see the three radar graphs together, which represent stress behaviors displayed during baseline, during the mount, and during the dismount. And you can see that the highest frequency of a stress behavior, and that behavior was ears back, was greatest at the mount, followed by the dismount, and then followed by baseline. And the second most common stress behavior that the therapeutic riding horses showed was tail swishing, which also occurred most frequently during the mount, followed by the dismount. So these are fairly subtle stress behaviors, but in fact, they are stress behaviors. Okay.
So again, in this study, we measured heart rate variability of the horses while they were under the conditions of the baseline, the mount, and the dismount. And so in here, you'll notice that we actually have the group data, and then we have, so the solid line represents the group data for the RMSSD, which is a time domain measure. So the root mean square of the standard deviation. So that's taking the R to R wave. So beat, 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 and then taking a derivative, an arithmetic calculation of that to actually make sense of that measure. And then what it's representing for us is that it's showing the parasympathetic nervous system branch of the autonomic nervous system. And then we compared that against the frequency domain measure, which is the low frequency to high frequency ratio. And what that does is that, excuse me, it helps to give us a balance between both the parasympathetic and sympathetic systems. So it's not that we just have multiple counts that we're using here or multiple different ways of making sense. Each one of the measures, the derivatives that come out of the heart rate variability complex or the ensemble actually tells us something different about the parasympathetic and sympathetic contributions to the neurological control of heart rhythmicity and of cardiovascular dynamics. So it's very important. But what's interesting here is that we found that there's a lot of consistency in the measures of the low frequency to high frequency measure, and there's a lot of inconsistency across the activities for the time domain measures. So again, it goes back to that notion of our previous study where we started to look and say, under different kinds of conditions, there are different kinds of responses, and we need to be aware that there's not one size fits all when you're looking at this data. Okay. I'm getting carried away here. Okay, so then the other thing we looked at was we looked at salivary cortisol. So now we're starting to introduce this notion of putting a hormone into our ensemble of responses to the stresses of the horse. And in this case, again, there was no significant difference before or after the therapeutic riding program, even though individually there may have been differences within the cortisol responses of a horse on a given day to a given activity.
So all of this, and we still don't have the answer to our question. So we had to keep doing some analyses.
Sorry, yeah, okay. So what happened then is we started to say, okay, let's start to really look at some of these, let's take a look at some of these measures that were visual representations of what we were trying to make sense of. And in this case, we looked at the BlandAltman plot again. And in this case, we were suggesting what's happening to the measure of agreement for cortisol and low frequency to high frequency ratio at baseline. They are actually in agreement statistically. But even though they're in agreement statistically at baseline, they're also in agreement at the dismount. So in other words, the low frequency to high frequency ratio and the cortisol were doing the same thing. So now you had a cardiovascular measure and you had a hormonal measure doing the exact same thing under two different conditions, but the responses are not identical. Okay, so you can see the difference in the shotgun patterns here. And that's the beauty of using the data visualization. Even though the data might have a single estimate and be completely different, in the visualization, you can see that the data are meeting the parameter boundaries, but the image pattern is considerably different. Okay. So in this case, what we say is that there's agreement between, sorry, I don't know, sync here? Okay, so here again, we use this notion of the BlandAltman agreement to show what's actually happening in terms of the cortisol to the low frequency to high frequency ratio. And it shows that there is consistency between these particular systems, and especially under different conditions, as in mount versus dismount versus baseline. Okay, so we're going to jump over this one, aren't we? Because this is a really big slide here. Okay, so now we're into our fourth study, okay, which is, we're getting close to the end here. That's the good thing.
Okay. So as we're getting a better and better understanding of the stress responses, we continue to apply them to other studies. And as I said, I'm a large animal surgeon, so I'm trying to teach low stress handling to our vet students and to our other clinicians, but you have to convince them sometimes that the horses actually are stressed and it's not just bad behavior. So this study was to measure stress levels in hospitalized equine patients during various veterinary procedures. And the procedures were normal procedures, physical examination by vet students, getting on the scale, and having blood taken. And we did our similar data collection where we had heart rate monitors on all the horses during these activities, and we collected saliva for cortisol measurements, and we also used an ethogram in this case. It was and we modified the ethogram specifically to horses in the veterinary teaching hospital. So it was interesting that there was no significant difference actually in cortisol between activities or any of our HRV measures. And there was no significant difference in most of the behaviors, but in this ethogram, we had ear flicking, and it was the behavior that was significantly different. And it was the only behavior consistently displayed by all subjects during all activities. And interestingly enough, it was highly correlated with some of our HRV measures indicative of stress. So this is just the bar graph showing that ear flick counts were significantly different between procedures.
So Rita jumped in and asked the question, when is it best to take the cortisol measures? Are we okay to answer a question?
I think this is the wrong slideshow. Okay. Sorry, when is it best to take the cortisol measures?
Yeah.
Yeah, that's a really tricky question.
Oh, sorry.
Or it's not a tricky question, it's a tricky answer. We read the literature for horses, and we used, so in some studies, the salivary cortisol increases 10 minutes after the stressful stimulus. So we were trying to use 10 minutes after each of the initiation of the different procedures. But it certainly gets tricky, and we definitely need to study that more, because we don't really know if we were studying the cortisol levels from all the procedures together, which one of our colleagues called all the mucking about, or the individual procedures, because most likely the time that we were allotted wasn't enough for the cortisol to come back to baseline. So again, we're learning a lot in each of these studies, and that's a pretty tricky thing when you're having multiple procedures. The other thing is in the hospital, we've got equine patients, and we have to somehow stick to the schedule of the clinicians as well. So we couldn't really say we're going to wait 45 minutes in between each procedure, or they would have got fed up with us. So again, it's real world data, and we're trying to balance the ideal research study design with the real world. Yeah.
One other response, Rita, that you might be thinking about was the concept of the time of day. And so we actually did go back to our data sets, because some of the Joyriders events were actually Saturday mornings, and some of them were in the Tuesday evenings, and we didn't find a difference across time of day. There was no circadian rhythm effect in these particular horses when we did our analysis, right?
Yeah. Okay, so it was interesting because we had the confidence that the ear flick was, the behavior was correlated with physiological indicators of stress. Our graduate student on this project actually set up a stress scale where she used a range of the number of ear flicks to represent the stress, and we validated this with students with different backgrounds in equine behavior, and it had really good inter-rater reliability. So that was a scale again. This is a strong slideshow.
I think so.
Yeah.
Okay, go ahead. So now what?
So now what? I think that's you, Bill.
Yeah. So I'm just checking. The slide should jump ahead here, and it's wait. This is. Let me just. Can I stop something here for a second? I just want to do a stop.
Sure.
It means that we had a.
With our technical difficulties, we don't have the ideal slideshow.
We
So.
do though, but it's still in there. Where is that? Hang on, let me get out of that. I'm going to get into this. Hang on 1 second. All right, this is what we want to show you, because this is the punchline now. This is what you've been waiting for. I couldn't wait for to tell you this punchline. Honestly, this is.
Do we need a drum roll?
You're going to want more than a drum roll. You're going to want a barrel roll on this one.
40 here. Yeah, I should be at 40.
Okay, hang on a second. Wait a second. What is going on here? Wait, no, that's not it. Okay, so give me 1 second here, Pebbles. Honestly,
so we can answer. I can try to answer other questions while Bill works on that, if anyone has them.
Yeah, hang on 1 second here.
So what we're learning, really, I think we made it clear, but all of these, heart rate variability is pretty complex. And what we rather than saying it's really not helpful, we've decided to go the other way and see the best ways to analyze the data. And so we still believe that it's a useful measurement. And I really think as a researcher, the thing that's helped me the most is doing the analysis of agreements and correlations. Because that's really, you know, ideally, when I first started, I thought, well, we're going to have, you know, if the horse is stressed, then we're going to have time domain, RMSSD will be decreased, LFHF will be increased, cortisol will be increased, and the behavior scale will be high. And of course, that's not how it worked. So, but little by little, as then I say to Bill, how, you know, okay, what are we going to do next for analysis, since he's the stats guy? And we literally spend hours and hours looking at all this data in different ways. So do you have it?
I do.
Okay.
So I'm going to play off the PDF here that we sent to you earlier, Pebbles, because for some reason in our messing around, we lost our SharePoint. But so basically in this webinar, what we're trying to describe is our journey to develop a better understanding of the well-being of horses. And I want to thank Molly Sweeney for that terminology, because this is exactly what we've been trying to do. We had a great chat about, it was probably a couple of weeks ago now, where we talked about what we were looking at in terms of wellness and human wellness and wellness in our veterans, military and police veterans for PTSD. And so we started to really try to get a handle on the understanding of well-being. And this has been for about 5 years we've been doing this over these different studies. And in particular, we're looking at the mechanisms of stress in the equine participants and how these mechanisms manifest different kinds of behaviors. So one of the areas that I work on is in concussion research, and in particular in concussion research, I'm always looking at behaviors. And so the way we describe this, if I can just bring this up a bit there, behavior is an explicit activity by an individual as a response to a stimulus. And behavior measurements provide us with an observable scale for interpreting how an animal is processing their environment. And in particular, the psychophysiology of behavior can be described by Posner's 3-part attention network test. If you see, you can pull that up, the attention network model. And so when I'm studying concussion, I often use an example where I would do something like drop something. And so that dropping is really important, because in Posner's attention network model, it's made up of 3 parts. It's made up of an alerting, which is the dropping and noise, and orienting, where did that noise come from, and then executing a behavior. So we call that the executive function. And so this model that Posner presented, it would be about 40 years ago now, over 40 years ago, where he talked about this way of describing how people and how stimuli respond to different kinds of stimuli around it. Okay, so how the organism responds to the different stimuli around it. So horses are no different. So here's where we're going with what we've been thinking about, the stress response. We think we can apply Posner's 3-stage model to interpreting the responses of stress among horses. First of all, in the stage 1 of Posner's attention network model is alerting. And so horses are constantly vigilant, and they're constantly monitoring the environment around them, and they're constantly scanning for this alerting stimulus. In the second model, this is a beautiful picture because it really shows that, sorry, I'm too far ahead here, that when a stimulus is detected, the attention network system invokes the orienting network to identify the location of the stimulus. Where did that stimulus come from? What was that noise I heard? What was that? Was that a gate opening? Was that someone coming with food? Was that someone coming to hurt me? And so on. So that's the orienting phase of the Posner model. And then in the third is, once the stimulus location is identified, the attention network system invokes the executive functioning of the network to determine what action or behavior will be executed as a response to the stimulus. So again, that's the third level of Posner's model. And so we think that this is a really important way to look at how we bring together our HRV measures, our behavioral measures, and our hormonal measures. So for example, HRV to us, it provides a proxy, okay? A measure, an external measure throughout the different stages of the attention network system when it's invoked. In other words, it gives us a measure alerting, orienting, and executive function. The stimulus detection during alerting and orienting stages, that can cause cortisol release by the adrenal glands, because the release of cortisol is important. It's necessary. It's a protective mechanism for us as humans, and probably for equine as well, because it's a psychophysiological response to a stressful stimulus. Because cortisol, it prepares the individual for action, because it's a catabolic hormone. That means that it helps to produce energy for action quickly, okay? And then we suggest then that one of the most common ways to show well-being in horses is by demonstrating that they're not stressed. And so then we can suggest that if we combine all these different measurement principles, so the objective and subjective measures of equine responses to the stimuli, with the different measurement techniques, like introducing these concepts like the BlandAltman two variable method to triangulate, to bring a point together, and then mapping these numerical events of our real world data onto theoretical frameworks like the attention network system, it helps us to provide direction in establishing robust and precise measures of the psychophysiological response to stress. And that is all we have.
So it's a lot.
Any questions?
Well, we're happy to answer any questions, yes.
Rita had a question in the chat box. Do you feel that HRV is not useful in a moving horse while the equipment is present?
So this year we're actually going to, that's a great question, Rita, because this year we're actually going to explore portable electrocardiogram against the heart rate monitor of polar heart rate monitors, to see the consistency of two different devices and how well they respond under movement. So right now, I'd say the jury's out on the movement measures.
Well,
you know, I mean,
I
wouldn't throw it out.
Well, you have to be really careful. So in the literature, the higher the activity, the less reliable the HRV is with either modality. But well, we were happy to see that in our hands, the polar monitor, which we've been using, because we can take it places and it's easy to use, we feel is reliable based on our reliability study during stall activity and cross ties. And so you might have noticed that in our studies, what we've done is use baseline, which is in a stall, so we trust that data. And otherwise we collect the data during a 5-minute period of time where the horse is standing still. So even if they walk over to the scale to get weighed, you know, once they're on the scale, we can collect the data while they're standing still.
Yeah.
And when they're standing still in the stall, getting their physical exam, with the horses participating in equine facilitated psychotherapy, they were standing tied to the arena, or they were standing right while being groomed, or they were standing right after they had been walked. So we're pretty careful to collect our data, you know, when the horses are doing low motion activity.
Yeah.
And as Bill said, we're going to try to investigate that further.
Yeah.
But right now we're just careful to use it when we believe it is reliable.
Yeah. I wouldn't say, you know, we've only done, you know, 5 years worth of studies. I think there's a lot more study that has to be done. I mean, honestly, that's science, right? We have a very specific group of horses. We have very specific devices. Other people, I'd like to hear what other people find in that area, so that we can actually do some comparison across research communities and see what's happening. We know that we get mixed results in humans with heart rate variability. So Rita, I think it's a great question, because I think there's a need to continue this research, and not to say one, to me as a researcher, nothing is definitive.
Questions.
So yeah, sorry, more questions, go ahead.
Yeah, you have more questions. So Peggy asked, what's the difference between the stall and the cross ties data versus walking data mean?
Well, remember we saw the chart. So we had the graph. Yeah, Bill's going to pull it up.
Yeah, right here.
This was our study where we were looking at stability of the heart rate variability data across 2 different days during 3 different activities. So there was good consistency in the HRV measures during cross ties and walking, but not consistency during the walk. So yeah,
Who asked that question? I'm sorry.
Peggy.
Peggy. So Peggy, we got consistent data in when the horse was standing still, and we did see measurements like heart rate and our data, but not as strong in terms of its relationship. And we definitely didn't see it as strong in the walking activity retest under those conditions.
Does that answer your question? Or are we missing something?
I haven't. She hasn't responded. She
Yes.
said yes. Thank you. And then Sandy's asked, is there a noticed correlation between the ethogram behaviors and the heart rate values?
Correlation with what?
Ethograms and
Ethogram, yeah,
yeah.
ethogram behaviors and the heart rate values.
Yeah.
So the answer is yes, there were under certain conditions again. So let me just see if I can pull that up here, which is the best measure. So we looked at behavior. Let me show you. I hope I have it here. I do have a graph of that. Here we go.
It is cortisol.
We have it for cortisol, and we have the same kind of data for the behavior as well. Here I showed cortisol because I wanted to show a very distinct hormonal response of cortisol and behavior. But I can definitely send us an email, and I can send you a similar graph for behavior and HRV measures.
So it's all, we tend to pick out certain HRV measures. I like RMSSD for time domain because it represents parasympathetic and is probably the best representative of the parasympathetic nervous system. I always look at heart rate because I'm familiar with heart rate and everyone's familiar with heart rate. So we usually include that too. And then for frequency domain, we like to use LFHF because it is supposed to show you the balance between sympathetic and parasympathetic. And now more recently, we're starting to look into the nonlinear domains and learn more about those, which may actually be a key to understanding the HRV better.
It might give us more precision in terms of our actual estimates. And so
yeah. So anyway, so we have different graphs, but you might have noticed with some of the graphs where Bill did the BlandAltman plots of agreement, it's, we have 2 of them that we show are RMSSD and behavior or cortisol.
RMSSD and cortisol.
RMSSD and cortisol, and then LFHF and cortisol, because those are the measures we tend to pick out that seem most useful. So, yeah.
And I just want to point out here that agreement is not a correlation. It's actually looking at 2 distinct events and whether or not they're traveling in the same 3-dimensional space together. Whereas in correlation, that's basically looking at 2 measures relative to the mean of each group. Okay. In this case, I'm saying when Bill does this, does he also do this? And the answer is yes or no, right? And when Peggy does this and Peggy does that, are they the same thing? And the answer is yes or no. So in other words, are those 2 events in agreement? And it's not a correlation. It's not a mathematical association. It's actually, do the events travel in the same space in a graph sense? Other questions? Anything else?
I didn't see any other. I was trying to scroll through the chat. Did we get everybody?
Most of them were saying it was a great presentation and amazing, very interesting. Thank you. And then Elizabeth said she now has 7 new hypotheses that she wants to test.
Bring it on, Elizabeth.
Very good.
Maybe she needs to ask y'all or share those with
Yeah,
you.
good. Well, we really enjoyed this, and it's again, we're still not finished. We still want to look at the point care plots and how they compare to some of these measures, as Laurie was saying, because you know the standard deviations of the way. Just imagine this is like a swarm of bees, and what's happening to the swarm of bees relative to these responses, and those responses represent the intrinsic physiology and psychophysiology of the horse participant under these different kinds of conditions, and tremendously exciting to be able to do this kind of research.
So we don't have the answer, which is the best measure, but we're going to keep working on that and see if we can figure out the gold standard.
Mm-hmm.
Any other questions? You can ask it if you want to, instead of just putting them in the chat box. All right, well, thanks everyone for attending, and I'm going to go ahead and stop recording at this time. And thank y'all for your presentation.
Thank you
You're
very much for
welcome.
having us.
Thanks a bunch.
We'll look forward to seeing you live and in person at the next HHRF conference.
Awesome.
Okay, good night.
See you later.